What I want to do is talk to you about our work on gene therapy and vein graft failure. And this has taken a long time. It's taken about 15 years since I started as a postdoc working in the Bristol Heart Institute. And it's just coming towards clinical trial. We're much slower than Roger, and we haven't injected any patients yet, but we've been fully funded to do a trial. And I'll outline the rationale and the preclinical data to justify that approach. Just by way of introduction, my lab is really interested in vascular complications with unmet clinical need, and we have a range of vascular remodeling aspects. And this has been focused traditionally in gene therapy approaches targeted towards vein grafts. And as I said, I'll illustrate that to you. We're also working on cell therapy approaches in ischemic disease using human embryonic stem cell approaches. And I'll talk a little bit just at the end about some microRNA work in vein grafting, but we're also particularly interested, as has been mentioned, in pulmonary arterial hypertension and the role of microRNAs in that. So in yellow here, I'll just what I'm going to outline in this talk for you. So when I started this project as a postdoc, there were about 800,000 bypass graft procedures performed worldwide each year, and so it was a major clinical problem. And the fact is that when you're using autologous saphenous vein, which is the main tissue that's being used for patients who require multiple bypasses, then there's a serious problem that the patency rate in the long term, so the 10-year patency rate is about 50%, so that's fairly major. The patency rates with arterial grafts are much higher, but of course there's less material to perform this, and most patients are particularly sick and require multiple bypasses. So interventional cardiology hasn't quite depleted the number of operations worldwide, and actually it's a very steady number. There are about 500,000 of these procedures performed worldwide each year, and about 23,000 in the UK using autologous saphenous vein. So this is the clinical problem. This is a late vein graft failure, and this is caused by a vascular remodeling event that's caused by acute injury to the vessel, exposure to higher blood pressure, leading to this near-wintimer formation, as you can see here. And this restricts the lumen and the blood flow, and patients reappear in clinic. As I mentioned, this is quite an important point relating to this, is this is quite a chronic failure rate. And we're trying a gene therapy approach that's quite acute, and whether this will work in patients is a bit of an unknown factor. So why are we trying a genetic modification approach in vein grafts? Well, quite simply, the acute injury that you see in vein grafts has been very well characterized. So when patients are grafted with autologous saphenous vein, there are a number of early events that lead to dysfunction of the vessel. So as I've mentioned, there is some vascular injury by the surgical procedure, but also by exposure to higher blood pressure. So you have platelet and thrombosis, you have endothelial injury, and you have inflammatory procedures. This leads to endothelial dysfunction and intimal hyperplasia, which is then creates a pro-atherogenic environment, and you have accelerated atherosclerosis, which will take between 2 and 10 years rather than traditionally decades. So this is an accelerated form that leads to reocclusion of the vessel. So how might a gene therapy approach work in this setting? Well, of course, we have access to the tissue, so that's delivery has been mentioned as being a fundamentally important factor. Of course, when we have the saphenous vein, this can be potentially be modulated ex vivo before it's grafted into the patient, so a significant safety bonus, if you like. So we can target these various procedures, particularly the remodeling events, with the aim of trying to create a vessel that is less prone to atherosclerosis. And we can do this by trying to modify the development of the near-wintimer formation and or improve endothelial function in so-called engineered vein grafts. One of the critical things about this has been the choice of model. There are four or five available. So there's a mouse model of vein grafting, which we've used a little bit. I'll talk about this right at the end. There's a rabbit model, which is pretty good because you can also fat feed rabbits. There's a pig model, which used quite a lot because anatomically pig vessels that we're using, this is saphenous vein to large vessels, are anatomically quite similar to humans. And we have an ex vivo human model where we have tissue from patients who are undergoing bypass grafting that's excess to surgery that we can manipulate in the lab culture and assess the effects on neowintimer formation. So we've really focused most of our developmental work on the pig and the human model, although we have used the mouse. The mouse has a significant limitations that you have mass cell death very early after grafting, and this is repopulated from the bone marrow, which is something that doesn't occur in pig or in human. So we've talked and we've heard already about vectors. If I could use an AAV for this, I would, but none of the AAV vectors that we've tried actually work in this setting. So what we have is a clinical window ex vivo where we can deliver the virus. This probably is about 20 or 30 minutes in which we require to get very good cannulation and ex vivo gene transfer to the vessel. The two viruses that do work in this setting are adenovirus and lentivirus. Lentivirus works very well, but it's very, very difficult to produce it to high enough titers. It's a bit of a bioprocessing issue. We cannot produce enough to do any preclinical studies in complex models. AAV vectors don't work in this setting. The 30-minute perfusion or luminal dwell that we have access to in the sort of clinical window doesn't result in any gene transfer that we can detect. So we're really, and also the dynamics, as Roger's mentioned about AAV, taking a little bit of time to switch on, whether that's going to be favorable or not in the context of vein grafting is open to debate. So we've focused for a long time on adenovirus. Adenovirus has been used quite a lot in clinical studies. About a quarter of all gene therapy trials have used adenovirus. They've had some good times and some bad times. But we think this is a pretty good application for the virus itself. So the way we do this very easily, this is a bit of human saphenous vein that's access to surgery, and we can cannulate this very quickly, and all we do is infuse the virus into the lumen and let it sit there. We don't add pressure from the outside or from the inside, and we just leave it for a luminal dwell. So this is using an adenovirus serotype 5 vector, which is the most commonly used virus. It's been pretty well characterized, although I'll come back to that in a minute. And if you just use a reporter gene analysis, what you find is very good levels of gene transfer. You have to push the system. So this is using sort of 10 to the 10 PFU per mil titers of virus, but you get very good endothelial as well as smooth muscle cell transduction. So this is in one that's been infected with the high-dose adenovirus. This is the control. So it's quite important to note, this is in the human model where we take these bits of vein and you culture them for two weeks ex vivo. At the time we did this procedure, of course, all the cells that were infected were at the luminal surface. And after 2 weeks in culture, these are the very cells that will form the neowintimer that you see, which is akin to what happens in patients. So it's about the smooth muscle cell migration and proliferation that you see following vascular injury. So we're actually targeting to the right cells in the pathology using this virus, and that's why we're particularly keen on it. As I mentioned, we also use a large animal model. So this is the pig model. This is an interposition graft into the pig's neck. This is the carotid. Then you have a bisection here. This is the saphenous vein. That's been autologous, taken from the pig's leg. It's been cannulated exactly as I showed you on the previous slide. It's been infected with the same dose of adenovirus, and then it's grafted end to end, as you can see here, and you have pulsatile flow. So again, you get very high levels of gene transfer using this dose, and you see endothelial as well as smooth muscle cell transduction. Because we use a relatively high dose of virus, the virus actually leaks. It goes into the vessel wall as well as into the endothelial and subendothelial layer. So we get quite a good distribution of the virus. So we've worked on a few transgenes in this setting. The one that we'll take to clinical trial is TIMP3. So this is a metalloproteinase inhibitor, tissue inhibitor, metalloproteinase 3. We've worked on this for a long time. We've figured out pretty much how it functions in a gene therapy setting. So when we infect cells with the virus, the virus will produce and secrete, the cell will produce and secrete high levels of TIMP3. We use a cytomegalovirus promoter, pumps out tons of the stuff. So TIMP3 is a little different to the other MMP inhibitors. It actually binds to sites within the extracellular matrix. So from there, it elicits a couple of effects. One is very picomolar concentrations. It blocks smooth muscle cell migration which is one of the main phenotypes of neointimer formation. And that's through classical inhibition of metalloproteinase activity, so type 4 collagenase activity. TIMP3, when you overexpress it at very high levels, can also sensitize cells to death. This only really occurs at nanomolar concentrations, but it's due to the effects of TIMP3 on ADAMs, which are involved in cleavage of death receptors from the surface. So you, in effect, sensitize cells to lower doses of death ligands, and we characterize this process as well. So in the vessel wall, when you overexpress TIMP3, you definitely have this process, and at high concentrations, so in the subendothelial layer, you'll have elevated levels of cell death as well. To show this works in vivo, so these are, this is in human saphenous vein. This is looking at directly at metalloproteinase activity. So this is type 4 collagenase activity in a process called in situ zymography. So in a control vessel, where you see white, you have active gelatinase activity. So the cells are producing lots of matrix-degrading enzymes. These enzymes then chew the matrix and allow those cells to proliferate and migrate. And as you can see in the media, but also in the neointimer, you have high levels of this enzyme activity. If you overexpress either TIMP1, TIMP2, or TIMP3, then you pretty much ablate this. So we know, using the virus, using the procedure and the infection conditions, we can block metalloproteinase activity throughout the vessel wall. And this is because these genes, proteins are secreted, so you have bystander activity, while cells that are infected by the virus also have effects on distant cells. And we know from work in the human saphenous vein ex vivo model that this is actually sufficient to block neointimer formation. So TIMP2 and TIMP3 are particularly effective at this, and this is just EVG staining of human saphenous vein. And we can see here that this is a control neointimer formation. And this is pretty much wiped out if you overexpress TIMP2 and TIMP3 in this setting. We spent a lot of time then working on these viruses in vivo, and it works out that if you take this to the pig model, then TIMP3 is the only one that really works, and it's probably due to either its maintenance of location to the matrix, or its ability to induce apoptosis at the same time. So this is the data in the shorter term pig model. So this is at 28 days. This is the neointimer that you see in the uninfected or the mock-infected grafts. This is a control virus, and this is pretty important that we know that the adenovirus is quite a complicated virus, has lots of different characteristics. But if you overexpress a control virus that doesn't express the gene, you have no detrimental effect to the graft pathology. And that's obviously a really important consideration. But when you overexpress TIMP3, you get a really robust inhibition in neointimer formation. So this is in the short term at 28 days, and there's been a lot of work in the acute vascular setting using these sort of approaches, and it's been found that you can have catch-up effects if you look a little longer. So we've done a series of studies which are taking the pigs out much longer, using a high N number. So in 3 months after grafting, we still get maintenance of this effect. So this is quite an important phenomenon, because we've heard that AAV will last for a long period of time. Well, adenovirus is quite immunogenic, and actually it won't. So a month after these grafts are put in, the virus is completely gone. Okay. TIMP3 hangs around a little bit, because I mentioned it binds to the matrix. But what we're seeing here is there's no virus at all left in these grafts at 3 months, but we have sustained efficacy. So that's quite important in terms of taking this forward clinically, and whether one of the main points is whether the dynamics of neointimer formation in the pig graft model is akin to what happens in human. We'll have to wait and see, and we can't really answer that until we take this forward clinically. So this is the justification for that approach. One of the important considerations is actually if you're inducing cell death, well, what's the effect on the endothelium? That would be a bad one. TIMP3 doesn't seem to affect this. We've done a lot of studies to look at endothelial regrowth across vein grafts, and we see absolutely no difference. So TIMP3 has a propensity really to be very selective in its phenotypic modulation of smooth muscle cells, okay, as you can see here. One of the other major concerns is inflammation, and whether if you pop an adenovirus into a vein graft, it suddenly becomes a very inflammatory environment. Well, it's already an inflammatory environment, and we have characterized in quite a lot of detail the T-cell response in vein grafts, and we don't see anything detrimental. I've already shown you that having the virus there in the short and the long term has no effect on neowinter formation. We have a little bit of excess T-cell infiltration at the early time points post-grafting, but we don't think this is really too much of an issue at this point, and we don't see a mass induction in the inflammatory response that you see in these grafts due to the virus itself being there. There are other genes that also work in this setting. This is P53. NOGO-B is the other one that works. It modulates vascular remodeling. So we have 3 different transgenes that seem to work in this setting. We haven't looked at them all in the long term, P53 and TIMP3. We have NOGO-B, we've only tested 28 days, but certainly we have some options in the future. So where we're going with this now is we've been funded by the Medical Research Council, developmental clinical studies with co-funding from the Heart Foundation to take this forward to a phase 1 and phase 2 clinical study. Phase 1 will be dose escalation up to the envisaged efficacious dose, and then phase 2 is a design which is placebo-controlled, within patient placebo-controlled trial. So patients who are eligible will have 1 arterial graft, but they'll have 2 saphenous vein grafts. The saphenous vein grafts will be randomized to active treatment or placebo, left or right, coronary, and in between 120 and 160 patients with neointimal measurement by optical coherence tomography at 1 year. But that's going to take us a little while. We're in the process of making the clinical grade virus at the moment, and we'll move forward to phase 1 as soon as that's completed and we've got regulatory approval. At the same time, just as Roger's been doing, is looking at alternative approaches or improving, I'd like to just spend a little bit of time talking about some of the issues with the adenovirus and whether it will work or whether it won't, and where we have backup options. So 3 issues that I can see in terms of using it in the setting. One is that tropism of the virus, adenovirus 5, for the vasculature is very poor. In fact, the receptor for the virus isn't expressed at all in the vasculature. So can we improve that? Pre-existing immunity to AD5 is extremely high. It's been reported to be as high as 90%. Whether that's an issue in the vein graft setting when you're doing this ex vivo and implanting is a different question. But obviously for regulatory authorities, it's a point of contention. And as I've already mentioned, duration and expression, whether that is significant or long enough with a first-generation virus to elicit an effect in humans is unknown, and certainly it works in pig. So one of the issues has always been with adenovirus 5 is it's quite complicated, and it's not a simple infectivity procedure. The virus is very clever and will use different proteins to infect via different routes of injection. We spent a lot of time trying to fill in some gaps about adenovirus infectivity, where we've discovered over the last few years that the hexon protein, which is this blue protein here, rather than merely being a major structural protein, is actually the main protein that's involved in infectivity of the virus in the in vivo setting, and it actually binds through host factors. It binds very tightly to coagulation factor 10, and this mediates liver gene transfer. So this really makes it quite complicated in terms of the different capsid proteins that contribute to infectivity. So you have the fiber, the penton, this is the fiber, and this is the penton, and this is the hexon, all interplaying in terms of tropism of the tissue that you're interested in. So I just want to talk about these 3 things very quickly in terms of how we may improve those for future application to vascular gene therapy. In terms of the pre-existing immunity to AD5 in the general population, reported to be up to 90%. So we've tested 100-odd sera from patients who attend clinics in the west of Scotland, and we certainly have the worst cardiovascular rates in the entire Europe. So we have plenty of patients. Our pre-existing immunity rate seems to be much lower if you use a traditional assay, which is basically just in cell culture using serum from these patients. We get an inhibitory, about 35% of our patients seem to have pre-existing immunity to AD5, which is actually quite at the low end of what's reported from populations elsewhere. But still is a potential issue. We're very interested in moving to different adenoviruses. So the adenovirus family, the human family, is 55 different adenoviruses apart from Ad5. So there's a lot of rich resource out there. Some of them are quite well known, the subspecies B viruses, such as adenovirus 35. Lots of these bind to CD46. But many of the different viruses, there's very little known about them, apart from their native pathogenesis. The problem with that is a lot of people haven't looked at them at all, and there are very few vectors available. So we're working within a European consortium to try and improve our knowledge of adenovirus through vector design and infectivity studies. This is just one example. If you take those same 103 sera and you use a rare adenovirus, this is from a subspecies D group, and you look at the same patients, then the pre-existing immunity to these so-called rare viruses is, of course, much lower. And in fact, there's only one patient here that has anything like pre-existing exposure to this virus. So we can switch. The problem is we don't really understand much about toxicity, safety, or infectivity of these particular viruses. The other thing is duration of expression, and it's been well known that you can manipulate the genome to improve long-term adenovirus gene transfer by manipulating and by removing essentially the entire genome and just leaving the inverted terminal repeats. A few groups have done this. It's very difficult to make. So we've made some helper-dependent viruses, and we've put them into the pig vein graft model. So this just shows you that at 3 months, these helper-dependent viruses are still producing lots of transgene, whereas, as I've mentioned before, the first-generation viruses don't. So this would give us another option to improve the longevity of expression in the vein graft setting, should we require it. I'm not sure about the regulatory aspects of using these viruses. I'm unaware of clinical studies that have been approved using helper-dependent viruses to date, although perhaps there are FDA processes in process, but certainly not in the UK. Finally, what about tropism? Well, as I've mentioned, this is quite complicated. We can manipulate very easily. In about a week, we can change the fiber, change any amino acid in the fiber, the hexon, the penton. We tend to leave the penton on its own because this is required for internalization of the virus. If you eliminate that, then you, in theory, reduce internalization, although that's not always true. We generally concentrate on modifications of the hexon and the fiber to try and improve the safety and improve the infectivity. This is just one example. So as I mentioned, in human vascular cells, so this is primary human saphenous vein endothelial cells or smooth muscle cells. So this is the target cell population for our trial. So if you look at CAR levels, which is the primary receptor that fiber of adenovirus 5 engages, as I've mentioned previously, it's not expressed at all. It's expressed in about 2% of cells at best. If you look at CD46, which is a well-known other virus receptor for different adenoviruses as well as different viruses from different clades, then essentially you have very high levels. So wouldn't it be obvious then to try and switch the infectivity to CD46 from CAR? Well, we can do this very easily. This is just one example. This is a virus we've generated which has two modifications. The first one is this T-star modification, which is a couple of amino acids in the hexon protein, and that eliminates that liver gene transfer, so it blocks binding to coagulation factor 10. And that's a sort of a safety application. So if you use surface plasmon resonance, which is basically looking at protein-protein interactions, if you take adenovirus 5, it will bind to factor 10 very tightly. This is a very low nanomolar affinity interaction. If you just make those mutations in the hexon, you eliminate this. So as you can see here, so then if you switch the fiber from adenovirus 5 to adenovirus 35, and this is a very nice one that Andre Lieber's made that has a picomolar affinity for CD46, and this is adenovirus 35 on its own. You can see it binds to CD46. But when you pop that fiber onto this background, you can see you've switched the tropism of the virus away from this sort of liver tropic virus to one that will purely bind to CD46. So it's quite simple now to make these modifications, and we've got many of these viruses that we're characterizing. So as you'd expect, the binding and the transduction of these cells in primary culture is much better than AD5, both in the saphenous vein endothelial cells and also in the smooth muscle cells. So you can see much higher levels of gene transfer at the same input doses. So the theory would be that we could actually improve the uptake of these viruses into the vessel wall, with the major point being that the clinical window that the surgeons will give us is 20 or 30 minutes at the most. So we need high affinity uptake kinetics of these viruses, and if we can get the dose as low as possible, then the profile of toxicity will be better for regulatory applications. So we can also make therapeutic viruses based on these backgrounds, and this is the one expressing TIMP3. And then if you just use the same multiplicities of infection of these different viruses, you get much better profiles, you get very high levels of therapeutic gene expression compared to the non-modified control, as you can see here on this image. And when you start to look at things like migration and proliferation, the parameters that you see are much better. So these viruses, the profile of these viruses in culture at least, looks good. And certainly, if you use these viruses at pretty low doses, so this is around 10 to the 9 input, you can get very good transduction with very short exposure times to human saphenous vein, and this is the clinical tissue that we'd also ultimately be targeting. So we can make these various modifications in the virus, and hopefully, in time, improve the potential of these viruses in the clinical setting. The last couple of slides, I'll just show you that we've not got all our eggs in one basket, and we're also trying to understand whether microRNA therapeutics would be useful in this setting. I think you'll hear a lot more about microRNA in the next talk, but also in the next day or two. We're quite interested because they've been associated with vascular remodeling in different settings. They haven't been tested as yet in vein grafting, so we're interested in whether this could be a therapeutic application. Quite complicated in where you would imagine that microRNAs would be involved, where we can focus certainly on smooth muscle cell migration and proliferation, but there are also many inflammatory components that may be influenced by microRNA. And I'll just show you a little bit of data on microRNA 21 to finish. So this is just immuno in situ hybridization using a control probe. I'm just looking in the pig model. So this is the pig model at day 28 after grafting in a control. So we have a lot of microRNA 21 in the media, but also in the neointima. And this is the human ex vivo model showing you quite high levels of 21 in those neointimal cells. And these co-localize predominantly with SMA. They've gone back to the mouse model to do this because we have the microRNA 21 knockout mice. In terms of vein grafting, this is a model that is good because you can do it in mice, but also taken with a couple of caveats that it doesn't really reproduce what you see in pig or human. But essentially, you do get this neointima formation. Okay, so this is a carotid model with the vena cava from a donor animal with this cuff over the end, so the bits of the artery are peeled back. And then you put the vena cava over this as a conduit. And you use different sections of this to look at neointima formation. It's an expensive model because you require 2 animals for every N of 1. This is just a profile of 21. This is just in the sort of donor tissue. This is the vena cava. This is the different arterial tissues. You have small levels of 21 in the arterial wall, as you will see here. But I think you'll appreciate quite quickly using in situ hybridization that microRNA 21 is activated quite extensively in this model and is localized in different areas in the media, but also in the neointimal regions quite predominantly. This can be quantified quite easily by qPCR, but we've done a series of studies to look at this in the 21 knockout mice, and what you find in the controls is this extensive neointima formation, which is blunted if you knock out microRNA 21. So this is using genetic ablation, not the anti-mir approach. I think the anti-mir approach might be quite tricky in the context of delivery, but we'll work on that in the future. So when you quantify this, then knockout miR-21 as a therapeutic aspect might also be useful in preventing neo-intimer formation. And maybe we need to combine some of our gene delivery approaches with cassettes that manipulate microRNAs in the future. I think that could be quite a rewarding area of research in the future. So just to sum up, I hope I've shown you, just summarised our work on adenovirus serotype 5. It's progressed towards a clinic. As I said, we're not as quick as others, but we'll be the first to do this in the context of vein grafting. Novel variants of AD5 and other rare viruses, I think, are likely to provide very fruitful avenues for enhancing gene delivery into the vasculature. And if we want to improve infectivity and safety in the long run, I think that can be rewarding, although I think we'll need to work with a lot of different people to make that happen. We can improve longevity, and maybe microRNAs are something for the future in terms of blocking vascular remodeling in the setting. I'd just like to thank everyone in my group, those people in red who contributed particularly. I'd like to, particularly to technicians that I have, that quite often don't get the credit, but these are fantastic people who do a lot of the core work, both cell-based and animal-based, as well as collaborators who have worked on this, long-term collaborators in Bristol and Hong Kong for the pig vein grafting model, microRNA with Eric Olsen and Eva Van Rooij. And particularly funding through the British Heart Foundation Medical Research Council, but also through a new Marie Curie training network that I coordinate that's been funded through the FP7 program, which is taking adenoviruses through to fundamental biology, but also through to clinical application in vaccine development, cardiovascular disease, and cancer gene therapy. And thank you for your attention.
Thank you very much, Andy.